Okay, we are now recording. Does anybody have any questions before we officially get started at 8 and before I do the announcements? All right, pretty easy. All 6 of you, well, yeah, there's only 6 of you, but anyway, attendance will be like normal today. Email me sometime before 9 a.m. to give me the keywords. Yeah, that's it for attendance, just normal thing. I'm waiting until 8 before I give you the first keyword because I want to keep it fair. Side note, well, it's 8 o'clock right now anyway. Anyway, I guess the first keyword is going to be sick because I am currently sick. So even though I had planned on being gone today anyway, it works out because there's no way I could have come in. I would assume it's not COVID. It's been not that long, but whatever it is, just in case it's contagious, I would not have come in today. So there's that. Anyway, again, first keyword for attendance today is sick. Can someone please confirm that you can hear me and see my PowerPoint?
I can.
Perfect. Okay. So to remind you with the pre-labs, the pre-labs that were due last Sunday, remember the big surprise this week was, hey, this week we're just doing the pre-lab lecture. So the pre-lab that was actually due last Sunday is now due this Sunday. So please, if you haven't done it, go ahead and do it. You'll get full credit. Well, you potentially could get full credit, assuming you get all the questions right. And if you missed anything, please go back and make your corrections because, again, you can get full credit for fixing your mistakes. And of all the labs we do this semester, this is the most important one as far as grades are concerned because you have to write a lab report on it. So you really, really need to understand what's going on. Also, if you were not in lab, you still need to do the pre-lab video and questions. Of course, at this point, it's too late for you to get full credit for that, but half credit is better than nothing. And again, you need to understand what's going on in this lab when you come in next week. So that video should help. And again, as always, come to me for help. So when you're working on the pre-lab, or hopefully you're looking at the lab before we come in next week, if you have questions, please come to me. I mean, some people are getting tutors, and that's great too. It's better to get help than do nothing, but definitely come to me if you can, because I'm the person who's grading, so I know exactly what I'm looking for. Any questions about lab stuff? All right, really quickly, just a real quick reminder for independent work. It's due Sunday like it is every Sunday. This week, you should be up to 36 points for an A. And again, if you need me, come see me for help. I can give you some ideas of what you can do for independent work. Any questions about independent work?
So if we come see you, do we just like stay in this or go to the meeting?
Yeah, there's a link that I've shared before, but I'm glad you've asked. I'll share it again. Basically, if you can't find the link, just email me and say, hey, I'm trying to meet you online. Can you send me the link? And I'll do that.
Thank you.
Good question. No, thank you. I'm going to also remind you guys, don't hit the submit button on pre-labs or independent work. Just do it. I've learned it is definitely automatically shared with me. So all you need to do is go into those forms I provided you and do the work. You don't hit the submit button. Anyway, going back to the question we just got, the great question we just got, office hours. They're going to be online today because I'm sick and because I'm not there. And unfortunately, I have a department meeting at 10. So I will be done with office hours at 10. However, as soon as the department meeting is over, I will come back on to office hours. So if you guys need me, just send me an email, let me know, and I'll find a time to get online for you. It's a good time to remind you about this 5% boost for anything you've done, whether it be the exams or labs, or maybe even feedback or independent work. That one might be a little bit harder, but if you meet me online, you will get a 5% boost for whatever assignment it is that you're letting me give you feedback for. So are there any questions about that? All right. That last bullet point is obviously just a joke. If you didn't get the joke, just look a little bit harder and think about it and you'll get it. Anyway, here we go. We left off by finishing up the overview of cellular respiration. And before we start jumping into the details, I'll remind you how the exam is going to go. And I'll also remind you that cellular respiration is made up of 3 stages. And most of the exam for chapter 6 is going to be like asking you which of the 3 stages. So for example, I might say which of the 3 stages uses oxygen, which of the 3 stages produces water, which of the 3 stages produces pyruvate, which you'll learn about here in a second, which of the 3 stages uses glucose. So as I'm going through all this, keep in mind that that's how most of the exam is going to go. Yeah, so here we go. Let's jump into it. So again, stage 1 is glycolysis. As you're studying, my suggestion is make sure you've memorized the stages in order before you start memorizing the stuff that I'm about to tell you. Make sure that you understand stage 1 is glycolysis, stage 2 is citric acid cycle, and stage 3 is electron transport chain. Once you have that memorized, then start going back and studying the stuff that I'm about to give you and memorize this.
Is your PowerPoint supposed to be changing?
Yes, it is. Thank you. Is it not?
No.
Thank you very much. Let me try something different. Excuse me. Okay. Present now, entire screen. We'll try this. Okay. And for that, on that note, the second the second word for attendance is going to be PowerPoint, since I am fighting with the PowerPoint. All right. And Vance, when you send me your email for attendance, please remind me that you brought that up, and I'll give you a little bit of extra credit. Should have done that yesterday when the other person did that, but I wasn't thinking, so I apologize. Anyway, glycolysis. Basically, glycolysis is all about breaking down glucose so we can get some high energy electrons. That's the basics, right? That's if you know, if you remember anything about it, remember that we are breaking down glucose to get high energy electrons. That's the big picture. Now, let's get a little bit specific. As you already know, glucose is C6H12O6. So as your book points out, glucose is a 6-carbon molecule. So what happens in glycolysis is we're breaking that in half. And I know none of you are math majors because I've looked it up and I've asked you, but can anybody tell me what 6 divided by 3 is? Excuse me, 6 divided by 2.
3.
Yay, exactly. So if you take a 6-carbon glucose and break it in half, you're going to get a 3-carbon molecule. And in this case, that 3-carbon molecule is called pyruvate. And you do need to know this for the exam. This is like 2 test questions right here. The first test question is, which of the 3 stages uses glucose? The answer is glycolysis. Another question could be, which of the 3 stages produces pyruvate? And the answer is glycolysis. Another one, and I probably won't ask it this way, but I might say, what happens when you split glucose? And the answer is you get 2 pyruvates. Another thing to point out about pyruvate before I move on to this next bullet point. That's really hard for me to write like this. So I'm only going to write this one word. If you read this, I don't know what your textbook says, but if you were to look this up in another source, you would see the word pyruvate, and you would also see the word pyruvic acid. And those things are the same thing. It's the same molecule, just a different name. Now, I use the word pyruvate instead of pyruvic acid, and you'll see why later. I do that to make things easier for you. So if you're taking notes, go ahead and write a little side note and say pyruvate equals pyruvic acid. So anyway, yeah, that's the first bullet point. Are there any questions about the first bullet point? Okay. Again, like I said, the big picture here is in glycolysis, you're breaking down glucose, and the whole purpose is to strip away high energy electrons. So that's where we're getting at in this bullet point. Because there are, well, obviously electrons in that molecule, and they are very energized. So the whole purpose of glycolysis and the citric acid cycle is to harvest as many of these high energy electrons as possible, because they're going to be used on the third step of cellular respiration. And to remind you from what we talked about on Wednesday, the third step, that's the moneymaker, right? That's the one that's making the most ATP. The first two stages or steps, however you want to say it, they're basically there to support the third step. And their way of supporting it is getting these high energy electrons. Now, let's get a little bit more specific. How are we moving these high energy electrons from one place to another? Because remember, glycolysis happens outside the mitochondria. Oh, this is so hard to do that. So I'll just put out M. It happens outside of the mitochondria. So we've got to get those electrons from one place to another. How do we do it? Well, we have this nifty little molecule right here. It's called NAD+. As far as you're concerned, it's just something that carries electrons. And when it has the plus in front of it, remember, electrons are negative. This thing's a plus. So hopefully that would help you remember that thing is ready to take on an electron, right? Negative things are attracted to positive things. So yes, this thing's ready to take on electrons, specifically 2 electrons, but I'm not going to be that specific on the exam, so I don't even care that you know the numbers. Just know that when you see NAD+, it is ready to move electrons. Now, when it gets its electrons, it is then technically called NADH because it also picks up a hydrogen. But you don't need to know that either. As a matter of fact, I'm probably not even going to ask you the difference between NAD+ and NADH. The point is, when you see any molecule that looks like this, NAD+, NADH, there's going to be some other ones I'm going to teach you about soon. If they look anything like that, your brain should think, electron carrier. That's the thing that's moving electrons around. That's it. Because remember, the first 2 stages is all about stripping away high energy electrons from that glucose and being, using those high energy electrons will be used at the third stage. So are there any questions so far? Okay, your book points out that glycolysis produces 4 ATP directly. And I don't care that you know that. That's why I'm putting an X in here. It's not because it's not true, but because I'm trying to keep things easy for you. What you need to know about glycolysis is it produces a little bit of ATP, just like the next stage produces a little bit of ATP. So the test question will be, which of the 3 stages produces ATP? And the answer is all of them. And then conversely, there'll be one that says which of the 3 stages produces most of the ATP, and that will be the electron transport chain. So again, glycolysis does produce a little bit of ATP. And to remind you, that's what all of cellular respiration is about, right? The whole purpose of cellular respiration is to get ATP. Anyway, glycolysis itself is broken down into 2 stages, 2 phases, as your book puts it: energy investment and energy harvest. Historically, I have not required you to know that for the exam. This semester, I'm not sure if I'm going to or not. So when you're studying, go ahead and keep that in mind, but I don't know. I probably won't ask you. Maybe it'll be an extra credit. I don't know. Anyway, the next slide I'm going to show you pretty much just visualizes what I've just told you. First of all, like I said, glycolysis happens outside of the mitochondria. That's this bean-looking thing right here. That's the mitochondria. So this all happens outside of the mitochondria. And again, the purpose here is to strip away electrons. And these electrons carried by NADH are going to go from glycolysis into the mitochondria where they're used later in the electron transport chain, which we'll talk about later. But also keep in mind, like I just told you, the glycolysis, you started with glucose and you broke it in half, and now you have these 2 pyruvates. And those things are almost ready to go into the mitochondria to go to the next stage. But we'll come back to that. Again, you start with a 6-carbon glucose, you end with 2 3-carbon pyruvates. Because, like I said, you're breaking it in half, right? So if you break that thing in half, one goes this way, one goes that way. Some other things happen that we don't talk about in this course, but yeah, that's what you end up with is pyruvate. Again, glycolysis is broken down into 2 phases. The first phase is energy investment, meaning you got to put a little energy in there to get this whole thing started. So specifically, you start with 2 ATP. I'm definitely not going to ask you that. If any, like I said, I probably won't even ask you about energy investment. But if I do, just know that just means that you're going to spend a little bit of ATP. But your book is very specific here. So this 2-ATP is used to transfer a phosphate group to that 3-carbon molecule that we were talking about, those 2 3-carbon molecules that we were talking about. But you don't need to know that detail. That's why I'm putting an X here. It's not because this stuff doesn't happen. It's because I'm not going to ask you about these phosphate groups. Anyway, once these things have the phosphate group, here's the part that you do need to know about. We are stripping away high energy electrons. And the thing that's moving and stripping away these high energy electrons, that's the NAD plus that's stripping them away, right? And once it has them, technically it is then called NADH. And again, as far as I'm concerned, those things are equal. Obviously they're different, but they're serving, it's the same thing. It just gets a different name once it picks up the electrons. Your book also points out that we have some more phosphate groups being added on, but I'm not worried about that detail either. So again, for this slide, I think what you need to know is that we break down glucose and strip away high energy electrons in the process. That's it. And sorry, one more thing, like I mentioned earlier, we do make a little bit of ATP. So any questions about this slide? This is basically glycolysis in one picture. And it's very specific. And remember, this is why I was, if you're taking notes, if you're a note taker, this is the chapter to take notes, because I do a lot of this where I tell you, don't worry about these details for the exam. So yeah, anyway, if there's no questions, we'll move on to the next stage. Oh wait, yeah, one more picture. Your book, I think, talks about how specifically we get ATP from this process. And your book talks about the fact that there's an enzyme that takes away one of these phosphates that I mentioned earlier, and it attaches that phosphate to the ADP. And when you do that, then you have something called ATP, which is what we're after. And obviously, since you took a phosphate away from this one on the left, then you're left with this molecule on the right. But this whole slide, don't worry about it. Not on the exam. You could write about it if you want for independent work. What is this enzyme that does this in glycolysis? What is this process called? You could look that up. Here's another thing that your book doesn't talk about and I don't talk about, and those videos I shared with you don't talk about. How is ADP made? That's a good independent work question, because you learn this whole thing about cellular respiration is how we go from ADP to ATP, right? We learned about how we add a phosphate group to ADP to make ATP, but where in the world did ADP come from? Anyway, here we go. That's it for stage 1. Are there any questions about glycolysis? The thing that starts with glucose and ends with pyruvate, and we stripped away high energy electrons and produced a little bit of ATP. Okay, now then, we are in stage 2. And again, before you worry about the details, the first thing you need to know or memorize is the fact that stage 2 is the citric acid cycle. And I'll also remind you, this is also called the Krebs cycle. And also, I will remind you, since I said this on Wednesday, the reason I call it the citric acid cycle, well, I'll come back to that. But yes, either one of these names are okay. They're the same thing. Anyway, the citric acid cycle takes the products of glycolysis, right? That's what we need. We broke glucose down in glycolysis, and that left us with pyruvate. And now that pyruvate, or those pyruvates, those are the things that are going into the citric acid cycle. However, for whatever reason, and you can look this up if you want for independent work, for whatever reason, the pyruvate itself cannot get inside of the mitochondria. So for that reason, it has to be groomed or converted to something else. And that's what we're going to talk about here. In the process, and remember, pyruvate, it's a 3-carbon molecule, right? It's got these 3 little carbons is the way it was drawn in your book. Pyruvate is a 3-carbon molecule, but to get it ready for the Krebs cycle, we lob off one of these carbons, right? So one of those carbons goes off as CO2. Now, you don't need to know this much detail. You don't need to memorize the fact that pyruvate is 3 carbons, and then we're going to lose one of the carbons, and then we're left with a 2-carbon molecule. You don't need to memorize that. What you do need to know, well, the test question will be, is which of the 3 stages produces carbon dioxide? And the answer is the citric acid cycle. And this goes back to why I call it the citric acid cycle instead of Krebs, because I want to make things easier for you. And if you can memorize, all these C's go together. Citric acid cycle, carbon dioxide, right? It's all together. So in the exam, when you have any questions about carbon dioxide, whether it's being produced in respiration or consumed in photosynthesis, the answer is always going to be some kind of cycle, because all these C's are going together. Excuse me. Anyway, any questions so far? Oh, do you have a question, or was somebody just accidentally hit something?
I was gonna just verify that you said it produces carbon dioxide, right?
Yes.
Okay.
Yeah.
Okay, thank you.
Citric acid cycle produces carbon dioxide. Or I guess a better way of saying that is the citric acid cycle of cellular respiration produces carbon dioxide. Because the big thing you need to memorize before these details is the fact that respiration produces carbon dioxide. That's the first thing you need to memorize. Once you have that memorized, then remember, all right, in which stage is carbon dioxide produced? And the answer is citric acid cycle. Now then, like I said, pyruvate is 3 carbons. We just lost one of the carbons. We lobbed it off to make carbon dioxide, and now we're left with this 2-carbon thing right here, and that is called acetic acid. Now, again, I'm not going to ask you these details. You don't need to remember that we had 3, lobbed 1 off, left with 2, and that 2 thing is called acidic. What you need to know, the way the test question is going to go, is which of the 3 stages is associated with acetic acid? And the answer is citric acid cycle. And now is my time to tell you why I call pyruvate instead of pyruvic acid. It's because in this exam, when we're talking about respiration, when you have any questions about acid, it will be the citric acid cycle. See how I did that? Nice and easy. And of course, if I called pyruvic acid, we couldn't do that. So in this exam, if it's about carbon, it's about the citric, or it's about a cycle. If it's about any kind of acid, it's going to be the citric acid cycle when we're talking about respiration, not photosynthesis. Any questions so far? All right. Here's the more important part as far as the process is concerned, not necessarily about the exam. But remember, like I keep saying, the first 2 stages of cellular respiration are all there to support the third one, because the third one is the moneymaker. That's the one producing the most ATP. And how does it do it? The third one uses high energy electrons. So the first 2 stages, it's all about stripping away high energy electrons, right? So it's still happening here. Just like we did in glycolysis, citric acid is doing it too. It is stripping away high energy electrons. And look, it's the same molecule. It's the NAD plus that accepts it, and that becomes NADH. Same thing, right? Again, a great test question. Which of the 3 stages strips away high energy electrons? And the answer is glycolysis and citric acid cycle. Any questions so far? All right. Here's where it gets a little bit more complicated. But don't write this down yet. Let me talk about it, and then you can decide what you want to write down. Like I said, we start with pyruvate, that 3-carbon pyruvate. We lob one of those carbons off. Now we have this 2-carbon acetic acid. And even that 2-carbon acetic acid, it's still not ready to go into the mitochondria. So we need to do a little bit more stuff to it. So each one of these acetic acids is then attached to something called coenzyme A. Now, when we're talking about illegal immigration, I know that sounds weird. I feel like you might think I'm going crazy, but hear me out. If you're talking about illegal immigration, does anybody know what a coyote is? Yes, not?
Yes.
Oh, what's a coyote?
That ain't like it's like a little, I don't know, a wolf.
I see you guys are giving me the
Is it-
correct, the actual answer. But if we're talking
Is
about
it the person that smuggles them?
yes, yeah,
Oh,
yes, you are correct. Somebody said wolf, and that is the correct, or a more correct biological answer. They are a wolf-like creature. But when we're talking about illegal immigration, a coyote is somebody that is hired to sneak in illegal immigrants into the United States. So I guess it doesn't matter since only one of you knew it, but maybe the one person who knew it, this could help you remember. Coenzyme A is kind of like the coyote, because acetic acid, it's not allowed into the mitochondria. So it's got to be snuck in, and that's where the coenzyme A comes in. It attaches itself to the acetic acid. That forms a new molecule called acetyl-CoA, and that thing is then ready to go into the mitochondria. All this being said, the only thing you need to know about this bullet point right here, the only test question there might be on this is I might say, which of the 3 stages is associated with coenzyme A, or which of the 3 stages is associated with acetyl-CoA? And the answer is the citric acid cycle. Any questions about this slide right here? All right. So technically, everything we've talked about is not really the citric acid cycle, right? Technically, we're talking about getting things ready to go into the citric acid cycle. But as far as we're concerned in this exam, everything we just said is part of the citric acid cycle for testing purposes. Because again, I might say which of the 3 stages is associated with acetic acid, and the answer is citric acid cycle. So that being said, let's talk about the actual citric acid cycle, which again, this is occurring inside the mitochondria. And here's what we just talked about. That pyruvate was broken down, right? We lost a carbon, that became acetic acid. We attached an enzyme to it, then it was called coenzyme A. At that point, we could sneak it across the border, and now it is ready to go into the citric acid cycle. Also, of course, the big, more important thing here is that we've stripped away electrons, and we've also done it here. Your book would be a little bit more accurate if it did that. But anyway, let's talk about the citric acid cycle. Oh, yeah, here's a big overview. Well, no, I'm sorry. Before we do, here's a visualization of everything we just talked about. Again, pyruvic acid. Let's call it pyruvate, remember, to make things easier. Pyruvate, which is what we got from glycolysis, it's broken down, right? We lose one of the carbons to carbon dioxide. That leaves us with acetic acid. Acetic acid is not ready to go, so not ready to go into the mitochondria. So we attach it to this coenzyme A, which leaves us with a molecule called acetyl-CoA. And again, for the test questions, that's going to go like this. Which of the 3 stages uses pyruvate? Not produces, but uses. Which of the 3 stages uses pyruvate? Citric acid cycle. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Which of the 3 stages is associated with acetic acid? Citric acid cycle. Which of the 3 stages is associated with acetyl-CoA or coenzyme A? Citric acid cycle. Which of the 3 stages strips away high energy electrons? And that would be glycolysis and citric acid cycle. Any questions so far? All right. So again, the whole thing about the first 2 stages, the citric acid cycle or glycolysis and the citric acid cycles, we're breaking down what we started with. We started with that 6-carbon glucose. It's got a lot of energy in it, and we'll learn about that in the next chapter. Every time we break it down, we're stripping away these high energy electrons. That's the main purpose of it. And in this case, as we're doing it, again, in the citric acid cycle, we're still producing more carbon dioxide. We're still stripping away more carbons. Your book gets pretty specific. So for every 1 molecule of glucose, in this particular portion, we have 2 molecules of carbon dioxide. Who cares? I don't care. What you need to know is that the citric acid cycle produces carbon dioxide. And again, like I said earlier, it does make a little bit of ATP, and that will be a test question because the test question will say which of the 3 stages produces ATP. They all do. Now, it's only the 3rd one that produces a lot. This one produces a little, and you don't even need to know how much. Just know that it produces a little bit. And again, more importantly for this process is this bullet point right here. It's all about stripping away those high energy electrons. And hey, here's that molecule I promised I would tell you about earlier, FADH2. Look at this. This thing, FADH2, just looks like this thing, right? When you're just looking at the words, they're both, and they just look similar. And They do the same thing. As far as you're concerned, they are the same thing. They just strip away high energy electrons. There's even one that we're going to talk about next chapter when we talk about photosynthesis, and it looks a lot like these. So anytime in this exam, when you see any word that looks like this, you should think, yep, that's the electron carrier. And I'm not going to try to trick you. I'm not going to say like, which one carries electron, which one is the electron carrier? And I give you, I don't know, CADH, right? Because that would look a lot like one of those. That would be tricky of me, but I'm not trying to trick you. So if you see any molecule that looks like this, even if you don't remember it and think, is that the one for sure? Don't worry, I'm not going to try to trick you. So if it looks like that, that is the thing carrying electrons. Your book also points out that all the carbon atoms that have gone into the citric acid cycle as fuel, specifically those 2 carbons from the acetyl-CoA, that is accounted for as carbon dioxide, and then a 4-carbon acceptor molecule that has been recycled. And I'm putting a big X through that because you don't need to know it. Again, for the exam, citric acid cycle produces carbon dioxide. It produces a little bit of ATP. And most importantly, it strips away high energy electrons. Any questions? All right. The next word for attendance will be the recommendation that I got at the end of the last lecture, which was donuts. Donuts, like the things that Homer from The Simpsons love. And I guess they make fun of, I guess the stereotype is that police officers like donuts. Anyway, here's a visualization of everything we just talked about. This acetic acid goes into the citric acid cycle. It's just broken down. The important thing here, again, we make carbon dioxide. This is the only stage that makes carbon dioxide. It makes a little bit of ATP, just like glucose makes a little bit of ATP. The most important thing here is that we're stripping away high energy electrons that are going to be used later, just like we do in glycolysis. Yeah, so that's it. And again, your book gets pretty specific. You can see here that there's this 4-carbon acceptor molecule, right? So this 4-carbon acceptor molecule hooks up with this acetic acid. 4 + 2 is 6. That new molecule is called citric acid, hence the name citric acid cycle. And then that thing is broken down between here and here. And in the process, again, that's where we're getting our carbon dioxide. Notice we're getting 2 carbon dioxides because here we have a 6-carbon molecule. Here we have a 4-carbon molecule. So we've got to lose 2 of them, right? And each one of them goes on to make their own molecule of CO2. But again, those are details you don't need to worry about. So are there any questions about this slide? This is a good time to remind you, I posted a video yesterday it was the crash course about photosynthesis. And technically you have to watch that before you come to lab anyway. So I would recommend watching it and doing the questions. But what I wanted to remind you after looking at this is that video, he goes into a lot of detail about the citric acid cycle. So I'm glad we've had this lecture now. So now when you watch that video, you can just keep in the back of your mind that when you see details that I didn't mention, just ignore it. Unless you want to learn it. Learning's great, but when it comes to the exam, you can ignore all those extra details. All right, so are there any questions about citric acid cycle or glycolysis for that matter, about the first 2 stages? All right, here we go. Like I keep saying, stage 3, electron transport, aka electron transport chain. This is the meat and potatoes. This is the money maker. This is where we make the most of our ATP. The other 2 stages were all about supporting the electron transport. Before we get into exactly what happens, let's talk a little bit about electrons, because your book does point out that during cellular respiration, we're talking about the big picture here, not necessarily just stage 3. When we're talking about the whole process of cellular respiration, what's happening is these electrons that I keep talking about, they're basically falling, meaning they're going from high energy to low energy. Because I keep saying in the first 2 stages, I kept saying strip away high energy electrons, stripping away high energy electrons. Notice I never said we're stripping away electrons. I said high energy electrons. And of course, we don't have time to get into the difference between high energy and low energy when it comes to electrons. It doesn't matter. Just know that they have a lot of energy when we strip them away. And what happens is throughout this whole process, they're losing their energy. And as you should remember from chapter 5, energy cannot be created or destroyed. So in this case, as that energy is being lost, it's not just disappearing either, it's being put to use. So keep that in mind as we talk about stage 3. Now, here's the part. Again, I'm just still trying to give you a little bit of background information before we even get into stage 3. I'm sure you guys have jumped cables before. Now, imagine, the other end of these, what are they, leads? Good grief. I'm so tired and sick right now. I can't think. Jumper cables. The other end of these jumper cables, let's imagine that's attached to the car that's dead, right? So this guy or girl, whatever, the person with these things needs to attach it to their car, the one that has the good battery. So what do you think would happen if they just attached the red one and didn't do anything with the black one? Would you be able to jump the car that way?
No.
That is correct. You would not be able to. And the basic answer, and it gets a little bit more complicated than this, but this explanation works for our purposes. The basic explanation is electrons have to have somewhere to go. So in this case, if you plug this up to this left one to the battery, electrons, they could go there, but since this black one isn't attached to anything, it's like a dead end. So the electrons just aren't going to go in there if there's a dead end. But once this black one is attached, the electrons have somewhere to go, and they flow all the way through here, through the dead battery, the dead car, and then they come back and go here. That's basically how it works. Again, that's not a perfect explanation when it comes to physics, but for our purposes, it is good enough. So keep that in mind when we're talking about the electron transport chain, because there will be some test questions when you need to think about the big picture of these electrons. Electrons have to come from somewhere. In this case, in this analogy, they had to come from the battery, and they have to go somewhere. And in this case, they would go back to the battery. But keep that in mind. Electrons have to come from somewhere. They have to go somewhere. And I've even told you that. Actually, I've told you where the electrons come from. I just didn't say it using that wording. So can someone tell me in this process, I haven't told you where the electrons go yet, but where do these electrons come from? No one? No guesses? I've been saying it. I keep talking about stripping away high energy electrons.
The citric acid cycle.
I like it. You are correct. I should have just worded it more carefully. You kind of answered more specifically, had I asked which stage do these high energy electrons come from, then you would be half correct because they come from glycolysis and the citric acid cycle. But the basic answer is these high energy electrons are coming from glucose, right? We had glucose, we broke it down to pyruvate. We had pyruvate, we broke it down into acetic acid. We had acetic acid, we threw that into the citric acid cycle, acetic acid. We threw that into the citric acid cycle. We broke it down some more. We kept stripping away high energy electrons. So technically, first we took it from the glucose, then we took it from the pyruvate, blah, 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 blah. But as far as this exam is concerned, and you need to know this, these high energy electrons came from the glucose. They came from the glucose. Know that for the exam. That will definitely be a test question. Where did the high energy electrons come from? They came from the glucose. And that's a big important thing because this whole thing is about the electron transport chain, basically, because the whole purpose of cellular respiration is to make ATP. And it is the third stage that makes most of the ATP. Anyway, again, electron transport chain, it's got all these high energy electrons that came from these first two stages. High energy electrons. Okay, again, like I've said earlier, on that other bullet point, the overall effect of these electrons being transported is sort of like a downward trip, only meaning, they don't physically go, from up to down, from higher to lower. What happens is, what I mean by that is they go from high energy to low energy. Again, like I said, energy just doesn't disappear. So as it's giving off, as these electrons are giving off the energy, that energy is being put to use. But again, for the exam, you need to know this one right here. Where do the high energy electrons come from? They come from glucose. And then here's the new information. Where do they end up? They end up at the oxygen. So oxygen, using the analogy I was using earlier, it's the black jumper cable. Electrons have to come from somewhere. They have to go somewhere. Where do they come from? They come from glucose. Where do they go? They go to oxygen. Again, using my analogy of the jumper cables, the glucose would be the red jumper cable. The oxygen would be the black jumper cable. And these NAD pluses would be basically be the cable itself, right? Electrons are coming from glucose to oxygen, and it's these things. It's these electron carriers that are moving them from the glucose to the oxygen. So again, this is a very important slide as far as the exam is concerned. And again, I'm going to give you the questions that you're going to get on the exam. Where do the electrons come from? They come from glucose. Where do the electrons go? They go to oxygen. What happens when the electrons go to oxygen? That forms water. Which of the 3 stages uses oxygen? Stage 3, electron transport chain. Which of the 3 stages forms water? Stage 3, electron transport chain. And here's a little preview, a little something for you to know for later. Even when we talk about photosynthesis, oxygen and water, they always go together. And I'll explain why later. But in this case, again, the oxygen accepts those electrons and it becomes water. So when you think of oxygen for this exam, you should think of water. It's always going to be in the same stage. Oxygen and water, always in the same stage. We're going to talk a little bit more about electron transport chain, but are there any questions so far?
Yes, I'm taking notes, so I just want to be sure.
Okay.
You said stage 3 uses oxygen and forms water, or forms oxygen and forms water?
Great. I'm glad you clarified. It uses oxygen and forms water.
Okay.
In a good way, this is a good time to remind you guys, if you ever, if you're not with me and able to ask me questions, if you get lost in the details, and that was a detail, then you got to back up and look at the big picture. And the big picture is that we're talking, technically we're talking about stage 3 right now, but the big picture of respiration is that we break down glucose in the presence of oxygen. That's so sloppy looking. To produce water and carbon dioxide. And of course, what we're after is the ATP. So that was a great question you asked. But what I'm getting at is, if you had that question or anyone else has any question like that again, and I'm not here to help, take a step back, look at the big picture. Because your question was it forming oxygen or using oxygen? And if you remember from the big picture, cellular respiration uses oxygen. And the easy way to remember that is, even though we're talking about cellular respiration, just think about respiration, right? You breathe in to get oxygen. You breathe out to get rid of carbon dioxide. But also in the process, there's a little bit of water in there too. So great question. I'm so glad you asked that. Any other questions? Okay. And really quick, when you see this equation, and you will see it on the exam, it will never say GLU, GLU, short for glucose. It'll probably either say the full word glucose, or it'll say C6H12O6. The only reason I wrote that is because it's extremely hard and sloppy for me to write the way I'm doing it. So here we go. As I keep saying, the whole purpose of the first 2 stages is to strip away these high energy electrons. And this is a picture from your book. In this case, they said from food, and that is technically correct. We'll get into that later. But as far as we're concerned, that might as well say glucose, because that's what we're focused on. Strip away these high energy electrons from the glucose. They go to the electron transport chain, which is what we're about to talk about. And you can sort of see this a little bit better. But for now, this will work. And they literally go from one protein to the next. And as they move from one protein to the next, they're kind of going down in a sense, meaning they're going from high energy to low energy. And as they go from one protein to the next, they're giving off that energy, right? And that energy that they're giving off, it is used to make the ATP. And again, this is stage 3, right? This is where we're making the most ATP. So this right here, this process we're looking at right here, this is what's making most of the ATP. And soon I'm going to tell you how these electrons make ATP. But for now, just know that these electrons are giving off their energy, and that is used to make ATP. And again, look, your book is very specific. Each NADH is carrying 2 electrons, and those 2 electrons then meet up with half a molecule of O2 and 2 molecules of protons, or excuse me, 2 protons, and that gives you 1 molecule of water. I'm not worried about the numbers. What you need to know is that high energy electrons, forget the numbers, have been stripped away. They come to the electron transport chain where they give off some energy to make ATP. Electrons have to go somewhere, or they have to come from somewhere. Where do they come from? They come from the glucose. They have to go somewhere. Where do they go? They go to the oxygen. And when that happens, the oxygen forms water. And again, your book is very specific with the number of molecules and the fact that we're also adding protons, but don't worry about that. Just know that electrons go to the oxygen and that produces water. Any questions about this slide before we talk about this little chunk right here? All right. The next word for attendance will be tissue. Like the things I'm probably going to run out of today because I'm sick. Tissue, the things you wipe your nose with. Anyway, next slide. The proteins of the electron transport chain are built into the inner membranes of the mitochondria. You do not need to know that detail, but that detail will be important for me to kind of explain how this process works. I'm going to do a really horrible picture here. The mitochondria, as I mentioned before, has 2 membranes. So like a cell just has one, right? You've got your membrane. Here's the outside of the cell. Here's the inside of the cell, right? The mitochondria has 2 membranes. So here's you got the outside of the mitochondria, and there's words for this. There's you got the inside of the mitochondria, and then you have this space in between the two membranes. So what happens is, like it says right here, the proteins of the electron transport chain, which is what we're talking about in stage 3, they're all built right here, like little bitty dots in the inner membrane. They're not on the outer membrane, they're on the inner membrane. And again, that's a detail I'm not going to ask you on the exam, but it's a detail you need to understand, or you need to know to understand how this process works. So we've got all these little proteins on the inner membrane. Your book also points out that my picture is garbage because the membranes are highly folded. So instead of being this one little circle, it's actually, you'll see when I show you a picture, it's folded. And the purpose is, the more folded it is, the more surface area you have. Therefore, the more surface area you have, the more copies of the electron transport chain, the more proteins you can have embedded in the protein, right? Because if these proteins are embedded in the membrane, obviously the more membrane you have, the more proteins you have. So again, your book is just pointing out this whole structure to function thing that we talked about in chapter 1. But for the exam, what's important is that these proteins are in the inner membrane. And again, not because I'm going to ask you where they are, but because I'm about to explain to you how this whole process works. So are there any questions about this slide? All right. So here's the better picture. There's the outer membrane. Here's the inner membrane. Like I said, my picture was garbage. You can see that the inner membrane is actually really folded because, again, the more surface area, the more proteins. Because what this doesn't show is there's a bunch of little proteins, embedded on the inner membrane. And I also said that there's different words that I didn't talk about. Like these little folds are called Cristae. The inside of the inner membrane is called the matrix. The space in between the inner membrane and the outer membrane, well, it has a word too, but I guess your book doesn't talk about it. That's okay. Anyway, you don't need to know these names. Just know that the mitochondria has two membranes, and it's the inner membrane that has all these proteins in it, and these proteins are about to be important. Anyway, this picture on the left is a drawing. This picture on the right is actually a picture. That's an electron microscope picture of what a mitochondria actually looks like. So here we go. Let's talk about some more concepts before I tell you exactly how this works. Your book points out that the energy that is stored by the electron transport is a lot like the water behind a dam. And I don't know if you guys know how hydroelectricity works, but on the other side of this dam is probably a lake, right? There's a lot of water. It's just water built up. So what happens when they need electricity is they open up some gates, and then the water flows through the gates, right? Can you guys picture what I'm saying here? what maybe you can't, sorry, what maybe you can't see, let's look at it like this. There's a lake, there's the dam, maybe there's a little thing letting the water through. Water comes out here, and then there's a river down here, right? And the water flows this way. And when that happens, there's a little thing, there's a little turbine, and it turns and turns and turns, and that's the thing that produces electricity because the water pushes the turbine as it goes through. So let me ask you guys this: when they need electricity and they let the water through, how do you think that works? Do you think they can just open it up and the water comes through, or do you think they need a pump to get that water to go from this lake down into that river? What do you think? Pump or no pump to get the water?
I think no pump.
Yes, you're exactly correct. No pump, because it's just physics, right? That's just how nature works. It's just going to go downhill. It's going to go from here's the word you need to know, even though this is not exactly accurate for the hydroelectric dam. It is accurate for what we're about to talk about. It is going to go from this area of high concentration to this area of low concentration. There's no energy needed. All they do is open up those gates and that water flows, right? High concentration to low concentration. And the same thing is happening right here in your mitochondria, except it's not water and it's not an electric turbine. Instead of water, we have hydrogen ions, and you need to know that. You need to know that for the exam. Which of the three stages pumps hydrogen ions against their concentration gradient? So in that case, what we're talking about is, in this case, you would need a pump to get the water to go from here to the left to the right, right? If you wanted to get it from the river to the lake, right, that's going to take actually energy. You got to pump it because the water wants to go the other direction. Same thing here. Hydrogen ions are going to want to go in the other direction. So we pump them against their concentration gradient. And this will make more sense in the next slides. Like I said, as the water flows down with the hydroelectric dam, that turns a turbine that produces electricity. In this case, what happens is, as the hydrogen ions go back down their concentration gradient, instead of having an electric turbine, there's an enzyme called ATP synthase. And that is the thing that lets the hydrogen ions through, and that is the thing that's making ATP. But you probably already knew that because, like I said in Chapter 5, and like I said you need to know for the exam in Chapter 5, enzymes end with the suffix "ase," and also enzymes are named after their substrate or what they do. So you probably could already guessed ATP synthase is an enzyme that synthesizes ATP. Anyway, yeah, the rest of this stuff I'm going to talk about here in a second. What you need to know for the exam is which of the three stages uses the enzyme ATP synthase. The answer is electron transport chain. Another test question is what enzyme, or another way of asking it possibly would be, which enzyme is used to make ATP in the electron transport chain? And the answer is ATP synthase. And keep in mind, there's been other ATP produced in previous, in stage 1 and stage 2. And we really didn't talk about how, because in this class, it's not important. There was an enzyme involved in Chapter 1 that we talked about that I showed you, or excuse me, stage 1, but we didn't name it. You don't need to know it. So the only time I have named an enzyme in this discussion is ATP synthase. So it should be pretty clear that that's important. So here's a picture of everything we're talking about. Again, we've stripped away all these high energy electrons from glucose in the first 2 stages. And then finally, they make it here to the electron transport chain. And what we're looking at here is, again, the inner membrane. Excuse me. This is the very inside of the mitochondria. This is the inner membrane, because remember, it has 2 membranes. And what's not shown here is the outer membrane. Of course, it doesn't really matter. That's why it's not shown. Anyway, what we're doing here is, again, these electrons go from, as I already showed you, electrons go from protein to protein to protein. And as they're doing that, I kept saying that they're giving off energy. And now I'm telling you how that energy is being put to use. That energy, excuse me, from those electrons, it's used to pump hydrogen ions against their concentration gradient. And again, you can look at this picture. There's a lot of hydrogens up top. There's not a lot of hydrogens down here. And as you know from chapter 5, that requires energy, right? Because that is going against the concentration gradient. That is active transport. It's going to require energy. Where does that energy come from? It comes from those high energy electrons that we stripped away earlier. Excuse me. So again, now we have all these high energy, we have all these protons up here. Then again, H plus is the same as a way of drawing proton. Anyway, we have a bunch of them up here. We don't have many down here. What happens? Nature just does its thing. These hydrogen ions just naturally diffuse, right, as things do, from an area of high concentration up here to an area of low concentration down here, just like water going through a dam. And what happens is it goes through this enzyme, and that enzyme literally spins like an electric turbine would. And that is the thing that makes the ATP. So yeah, this is a very busy picture, but it's really not that complicated if you just step back and look at the big picture. And again, we've stripped away high energy electrons from the first 2 stages. We bring them, they go from protein to protein, giving off their energy. That energy is used to pump hydrogen ions against the concentration gradients, which eventually just diffuse back down through ATP synthase, and that's where ATP is made. Excuse me. And again, like I've already said, electrons have to come from somewhere. They have to go somewhere. Where do they come from? They came from the glucose. Where do they go? They go to the oxygen, and that forms water, right? So it's all right here, everything you need to know. There's also this little skull and crossbones. And this basically, if I remember correctly, your book points out that like a lot of poisons or toxins, some of them, the way they kill you is they stop this process right here happening. Or another way you could look at it, if you drown or you suffocate, if someone chokes you to death, this is how it kills you. It's because you don't have oxygen. Therefore, these electrons have nowhere to go. Therefore, these protons are not pumped against their high concentration gradient. Therefore, we are not making a whole lot of ATP. And your cells need ATP to survive, as we know from chapter 5. And without it, you die. So this is what kills you. This is how a lack of oxygen kills you. What else can I say? I want to make sure. Oh, yeah. Here's another. I'm going to put it next to this because it's a detail you don't need to know. Technically, it's the FADH2 that's moving these electrons, and you don't need to know that. Again, if you see anything that looks like this, you should think, oh, yeah, those things are just moving electrons. And I don't even care that you know how the electrons get from protein to protein. Doesn't matter. The fact is they're going from protein to protein, and they're giving off their energy. And that energy is used to pump hydrogen ions. Any questions about this slide? All right. This is good. We have enough time to do the results of cellular respiration, which is almost like a big review of what we just talked about. Again, like I've said before, you need to know this number. I'm not big on numbers, but you need to know this one. Cellular respiration produces about 32 ATP. And again, don't look this up because people don't necessarily agree on this number, and I teach out of the textbook, so I'm requiring you to know the number 32. Other sources will give you another number. I think the video I shared with you actually said 38. I don't remember for sure, but anyway, you're responsible for the number 32. And this is a good picture to study. It's a little bit more specific than you need to be. Yes, there's 2, but who cares? Yes, we've made 2 there. Where are we at? Who cares that we made 2 right there? Yes, technically we made it about 28 right there. Doesn't matter. What you need to know is glycolysis makes a little bit of ATP. Citric acid cycle makes a little bit of ATP. Most importantly, they are breaking down that glucose and stripping away high energy electrons that go to the electron transport chain where ATP makes the most, ATP synthase makes the most ATP. So was a lot. Was that a question somebody has, or is something happening? No, I only have 2 minutes. Anyway, there's a lot of test questions here. Which of the 3 stages happens inside the mitochondria? Citric acid cycle and electron transport chain. Which of the 3 stages happens outside of the mitochondria? Glycolysis. Which of the 3 stages strips away high energy electrons? Glycolysis and citric acid cycle. Which of the three stages uses high energy electrons? Electron transport chain. Which of the three stages produces ATP? All of them. Which of the three stages produces the most ATP? Electron transport chain. How many ATP are produced overall? About 32. Which of the three stages uses glucose directly? Glycolysis. Which one produces pyruvate? Glycolysis. Which one is associated with acetyl-CoA and acetic acid? Citric acid cycle. Yeah, there's a lot of it. So anyway, it's a good picture to study. Are there any questions about this picture? All right. Really quickly, I'll point this out. I keep saying glucose, glucose, glucose. We're breaking down glucose. We're breaking down glucose. I'm sure some of you guys had breakfast, maybe dinner last night, and you probably ate more than glucose. You probably didn't just eat glucose. So just want to point out really quickly that obviously, yes, you're going to break down other stuff than glucose. As you know from chapter 3, there's biological molecules. Sorry, guys. I'll end it there. My phone just skipped and it says it's 9 o'clock. Can somebody tell me what time it is?
9 o'clock.
Is 9.
Good grief. I'm telling you, I swear, I promise you, I've been looking at my phone and it hasn't been saying 9. It jumped to 9. Well, okay. I went over. And for those of you who are still here, let me do another keyword. Keychain is the final keyword. So anybody who doesn't have keychain will not lose points, but those of you who do will get extra points. I'm so sorry for keeping you over, but you will be rewarded with some extra attendance points.
Okay. Have a good weekend.
Thanks. You too. I'm so sorry for keeping you guys over.
Can you send the link again for the meet?
Yes. Can you email me?
Yes, I can.
And I'll just email it back.
Okay, thank you.
You're welcome.